Good morning and good afternoon again, everybody. My name is Avi Saharia, and I'm here, Director of Cell-Based Assays here at DiscoverX. Today I'm going to talk to you about our ready-to-use cell-based assays for the development of potency assays for anti-VEGF molecules like Bevacizumab, Aflibercept, and Ranibizumab. Before we get started today, I would like to make a couple of administrative announcements. This webinar is being recorded, and so you will get a copy of the recorded webinar as well as a copy of the slides shortly after the end of the webinar. If you have any questions at any point of time during the course of the webinar, please type those into the Q&A box and the chat box that you can find on the drop-down menu on the WebEx for this webinar. So with that, I'd like to go ahead and get started on the discussion on the use of our cell-based assays for development of potency assays for anti-VEGF drugs like Bevacizumab, Aflibercept, and Ranibizumab. I'm really excited to talk to you about this today because I think that this is something that has the potential to really accelerate the development of these QC lot release assays for your team and transform the way that you've been doing potency assay development. So the agenda of my discussion today is I'm going to first focus on some of the technical challenges associated with biosimilar development and then start discussing a potency assay that we've developed for Bevacizumab with some qualification data as well as validation data associated with that. And then dive right into some qualification data also with Aflibercept and Ranibizumab, both of which are anti-VEGF molecules which have a slightly different structure than Bevacizumab. And then finally touch upon the use of cryopreserved ready-to-use cells, and in particular the advantages that this sort of system offers to you by eliminating the need for cell culture in your facility. So when we think about some of the challenges associated with biosimilar development, we realize that the regulatory authorities, particularly the FDA and the EMEA, require that to demonstrate biosimilarity, there needs to be a demonstration of what's called no clinically meaningful differences between the innovator and the biosimilar molecule, which, as the FDA requires, is a totality of evidence to demonstrate this no clinically meaningful differences between the two molecules. And that, as shown in this schematic at the bottom here, requires a number of different steps to be performed, ending with human studies or clinical studies that show that these two molecules have exactly the same performance in a clinical trial. And a foundation of that, this process or the development of this or demonstration of interchangeability or biosimilarity is the development or the need for functional cell-based assays to demonstrate comparability in terms of functional performance between the innovator and the biosimilar, as well as the use of these assays further downstream in bioanalytical studies to demonstrate lot release testing for these molecules. And so as I mentioned, these assays, accurate and precise bioanalytical assays, are critical to establish comparability and as well as for development of lot release assays for these biosimilar molecules. So given their critical nature or their importance in the demonstration of biosimilarity, of course there exists a number of different assays that people have already been utilizing, especially because these drugs have been now developed. Most of the molecules that we are looking at as biosimilar candidates are candidates that were developed 10, 15, maybe even longer than that time ago, 10, 15 years ago or longer as drug molecules. The assays that exist for those are all assays that have utilized technologies from that time. So assays that exist that people have utilized for development of biosimilar molecules are based on animals or cell-based proliferation assays or primary human cell-based assays, and in some cases even radioactivity-based assays. And these assays have a number of different challenges. They oftentimes require a tremendous amount of assay development before one can even get it to a point that allows them to get to qualification studies in the lab. So this has a tremendous cost to the program of 3 to 6 months of lost time, and in particular, this is critical in the biosimilar space as there is a tremendous race to get your biosimilar molecule first to market. Oftentimes these assays, all of the assays that I've mentioned above, have long multi-step protocols, and these tend to result in provide results in 4 to 6 days per assay, which increases the amount of cycle time associated with each assay, increasing the total amount of time required for validation, increasing the total amount of time required to run each sample once that data or this assay has been validated, for example. Oftentimes, these assays tend to be very complex, which leads to high variability, particularly when we think about animal-based assays, proliferation assays, even primary cellular radioactivity-based assays. And these complexity associated with the assay leads to oftentimes, again, difficulty in precision and a number of different failures that might be seen in the number of runs with a particular assay. And then in particular, complexity of an assay, including the need for culturing cells for a long period of time before actually running the assay itself, oftentimes leads to difficulty in transfer of the assay from the site that it was developed to CROs, CMOs that might be running this on a more routine basis, or even global manufacturing sites for some of our big pharma clients as they transfer it out for clinical trials as well as for their marketed molecule. And one of the last challenges that I want to focus in on is the specificity of the assay readout. And if there is the lack of specificity, especially in scenarios where you're looking at a readout that is very far downstream of the actual challenge or the molecular challenge itself, is that oftentimes you're looking at the potential for matrix interference. And that complicates the assay, especially when you think about the use of those assays for neutralizing antibody studies in a further downstream in the clinical trial. So given some of these challenges that people face with biosimilar development and assay development for QC not release assays and potency assays, we sought to develop a system that would address a number of these challenges and alleviate this, enabling you to develop assays faster, have a shorter, easier protocol to read, as well as much higher reproducibility and easy to transfer globally. And to address those, I'm going to walk you through one particular example. And the example, as the title of this webinar indicated, is for anti-VEGF molecules. So there's 4 major anti-VEGF molecules that are currently in the market: Avastin, Eylea, and Zaltrap, both of which are Aflibercept, Avastin being Bevacizumab, and Lucentis, which is Ranibizumab. These 4 molecules that are currently out in the market are at various times in the next few years are coming off patent, and therefore there's a number of companies that are actively developing biosimilars or biobetters against these targeted molecules. And so one of the key points, as I mentioned earlier, when developing biosimilars or biobetters against these molecules is the need for functional assays. And so let's take a look at what's the clinical mechanism of action for any of these drugs. What we do know is that the VEGF molecule increases vascularization in either in tissues, and in this the disease tissues that people are looking at in these cases is either metastatic cancer or wet AMD in the case of Eylea Lucentis. So here we are looking at VEGF, which increases vascularization by activating and increasing the proliferation of these vascular endothelial cells. At a molecular level, however, what occurs is the VEGF-A molecule actually binds to the VEGF receptors and causes the dimerization of these VEGF receptors, which is an essential event in the activation cascade of this receptor. This dimerization event actually leads to then transphosphorylation of the VEGFR2 receptor, and therefore the downstream signaling cascade, which eventually leads to proliferation of these vascular endothelial cells. The binding of this VEGF molecule with a drug like Avastin or Aflibercept or Ranibizumab prevents the VEGF-A molecule from binding to and activating the VEGFR2, and therefore prevents it from dimerization and transphosphorylation, and of course all the downstream signaling events that occur after that. So as you can see that at a molecular level, the way this works is to prevent the VEGF-A molecule from binding and activating the VEGFR2. And this is exactly the way we've approached the assay, as you will see in a couple of slides further down. So what is the existing assay that currently people utilize for these anti-VEGF molecules? The existing assay for the anti-VEGF molecules that has been utilized by numerous companies that have developed all of these molecules in the past is the HUVEC proliferation assay. And the HUVEC standing for the human vascular endothelial cell proliferation assay. And this assay tends to be a very long, arduous, and difficult assay to run. So the way it would start, and I'm going to walk you through the process of this assay itself. This is all information gathered by discussion with a number of our colleagues who've been doing these assays for several years now, is taking these human endothelial cells, which are again primary human cells, thawing these frozen cells, culturing them for at least a week, plating the cells again in our assay plate, adding the molecules, incubating it for a period of 96 hours, which is 4 days, adding then Alamar Blue on the 5th day, incubating it for 6 hours and reading it. So you can see that the total assay time actually from once you've plated the cells to read it on a plate is about 5 days, and the cells require at least a week of culture prior to running the full assay. So the entire cycle time associated with this assay is approximately 2 weeks. And so the challenges that one can anticipate in an assay like this, as based off of our discussions with numerous colleagues, is that, of course, we've just talked about how long this assay is. So you've got reduced lab efficiency because it requires months to develop. You need to culture cells, so several hours of culture required in a GMP environment. It has a very long protocol, which of course reduces cycle time and therefore lab efficiency. But importantly, because the cells are required to be cultured before actually running the assay, there's a lot of questions about challenges associated with culture-dependent variability that one can observe in the system. And so therefore there's reduced consistency associated with this assay because of the need for these cells to be cultured, of course, and then also because these are primary cells, and so you've got associated donor variability that might come into play. You've got reduced flexibility because of the amount of time required to run each of these assays, difficulty to transfer the method to global sites because of, again, the complexity associated with handling and culturing these cells, and the associated cost of culture and operator time for this assay. And so because of the challenges associated with this assay for the HUVEC proliferation assay, as useful as it has been for the past couple of decades in enabling the development as well as the lot release of these drugs into the market, is something that we wanted to address. And to address this, we developed an assay using our proprietary PathHunter technology, which is the EFC technology. And what that really means is it's enzyme fragment complementation. And at its heart, it's a split beta-gal system. So we've taken the beta-gal enzyme, split it into two components, the enzyme donor and the enzyme acceptor. And when they're apart, they're completely inactive. However, when they come together, they create now an active enzyme, which can then hydrolyze substrate to give us a chemiluminescent output. Very simple system, very, very easy to utilize, and it gives us a chemiluminescent output which can then be read on any benchtop luminometer. So how do we use this simple technology to create an assay for the anti-VEGF molecules? So the way we do this is, again, rely on the molecular mechanism of action, or essentially the native receptor biology for these. So as I mentioned, and as I showed you earlier, that the VEGFR2 receptors homodimerize upon getting activated by the VEGF-A molecule, and this is an essential component or an integral part of the activation cascade of these receptors. So to interrogate this early event in the VEGFR2 receptor activation cascade, we tagged the VEGFR2 protein with the EA tag and another construct with the PK tag, which is essentially another, it's a fragment of our enzyme donor. When the VEGF-A molecule binds to and activates the VEGFR2 receptors, this leads to dimerization of these VEGFR2 receptors, bringing the enzyme donor and the enzyme acceptor into close proximity with each other. This forces them to then complement, creating then an active enzyme which can hydrolyze substrate and give chemiluminescence. Anti-VEGF molecules like Avastin or any of the molecules that we've mentioned earlier can bind to VEGF, prevent it from activating the receptors, and therefore we do not get any dimerization event of the receptors, and therefore no signal associated with that. So again, just to reiterate, when we get adding VEGF increases the amount of total light output, whereas adding our anti-VEGF molecules decreases the amount of total light output, as you will see in the upcoming slides in a couple of minutes. So altogether now, what we can see is that this assay, as simple as it is, again, allows us to interrogate the native biology of these receptors. And the protocol associated with this assay is something that is incredibly simple as well. So as I mentioned, all of these assays have been developed now and been adopted to a frozen thaw and use format. So in this case, you would actually take the frozen cells provided, thaw them directly into your assay plate, add in your agonist molecule, which is VEGF-A, as well as your inhibitor molecules, the anti-VEGF drugs, incubate overnight at 37 degrees, add your detection reagents, and read the luminescence on a benchtop luminometer. So you can see that the entire assay has now been reduced to less than 24 hours, less than 1 day to cycle through an entire Bevacizumab bioassay. Compared to the HUVEC proliferation assay, where if you include the amount of time required to culture the cells before actually running the assay, you can observe here that it takes 2 to 3 weeks to fully run through a HUVEC proliferation assay, and we've been able to reduce that down to less than a day with our PathHunter Bevacizumab bioassay. And that's actually really incredible because it reduces your cycle time, increases the efficiency of your lab, and allows you much greater flexibility where you can run the assays as and when the samples that you have start coming in. So the technology, as I mentioned, is quite simple. It has a fantastic assay performance, a very simple protocol and a very rapid result. How does the assay actually perform? So the rest of the webinar, I'm going to talk to you a little bit about data associated with this assay, including qualification data and validation data that we've done, both internally as well as with a number of our different partner companies. So in this case, the first piece of data I wanted to share with you is that if we have a dose response curve where we have an increasing amount of VEGF-A added to our cells, and we can observe a nice sigmoidal dose response curve, and on the y-axis we are looking at essentially the relative luminescence units indicating an activation of the VEGF receptors in the presence of VEGF-A. And as would be expected, in cases where we get increasing concentrations of the anti-VEGF molecules like Eylea, Avastin, or Zaltrap, we can expect to see a nice dose-dependent decrease in the total amount of signal present, again indicating a reduction in receptor activation or a reduction in the activity of the VEGF molecule, again all based on the mechanism of action or the true biology of these receptors. And of course including a control like cetuximab, which does not affect the activation or the inhibition of the VEGF molecules, leads to essentially a flat curve as would be expected, again demonstrating the specificity of the assay. So once we had observed that this assay does perform well with marketed drugs, a question that we wanted to ask ourselves is, how does this assay perform through a qualification experiment? And so to do that, we actually did some initial qualification exercise performed here at DiscoverX with our R&D team, where we looked at expected potencies of 70, 100, and 130% on 3 separate days, again, using a single analyst in this time. And what we observed was that we were able to get a good recovery and a good percent RST value associated with these assays. And this is again done with the plate layout shown on the right side, where we are looking at these assays being run in duplicate with the appropriate dilution series shown on the slide. So altogether, what that indicated is that this assay does suggest that it would perform well in a validation exercise. And so this actually allowed us to now work with some of our partner companies to start thinking about putting this through a full validation exercise. And so the approach we took was to utilize the validation criteria as per the ICH guidelines. I've already demonstrated to you that this assay is based on the mechanism of action of the drug as well as the receptors. And we started looking at some of the other criteria that are highlighted or that are suggested for based on the ICH guidelines for validation. And to do a full validation of these assays, we worked with one of our collaborators, in particular Covance Labs, where they have deep experience with development of bioassays for biosimilars, and they have deep experience in terms of developing end-to-end solutions for their clients for the development of biosimilars. And in particular, they have quite a bit of experience with biological assays or bioassays in this case. So Covance, our collaborators at Covance chose to utilize the USP 1033 biological assay validation protocol. And here's a brief summary of what that protocol looked like. We utilized a nested design with 2 different analysts targeting potencies ranging from 50% to 150%, and utilized 6 independent runs per analyst to achieve a total data set that was then utilized for analysis for full validation of this assay. And the assay actually then performed very robustly in this case. What I'm going to share with you in the next few slides is actually just data from the partial validation associated with that. I've worked now with our colleagues Paul and Stu at Covance, who've informed me that the validation has been completed, and they have not yet shared the full validation data yet with us because the report is still being finalized and signed off. However, what I am able to share with you today is partial validation data that they have shared with me so far. And as we can see, is that the assay performs very robustly in this case, where you're looking at a precision of about 98%, accuracy of 4.8% associated with this assay, indicating that it has a very high reproducibility as well as a very high and a very good recovery rate associated with this assay. And we've got examples of the constrained data as well as the unconstrained data demonstrating that the actual performance and the data sets itself themselves look very good in our assay, demonstrating good parallelism in these assays as well, and a good linearity associated with this assay. So altogether, it suggested that this assay, or indicated that this assay has been validated at Covance and can be validated at any other client sites for use in lot release testing of biosimilar or biobetter molecules. In addition to performing all of these robustness studies for validation, we also tested for specificity of the PathHunter assay. And in this case, as you can see, the assay only responds to Avastin as expected, and not to MabThera, which of course does not target this VEGF molecule. So one of the goals that we had set out initially to address with Covance was that we wanted to compare the performance of our PathHunter Bevacizumab assay to the HUVEC proliferation assay at Covance. So the team at Covance tried to validate and draw the comparison between these two assays. But however, as you can see, that in an early qualification experiment with the HUVEC assay, the HUVEC assay did not perform as expected and had a very, very low recovery percentage associated with this assay. So based on this data, we did not go through the full validation with the HUVEC assay and only completed the validation with the PathHunter assay, and as I noted, which is now complete as well. In addition to our partnership with Covance Labs, we also had a similar partnership with SGS Labs. And again, the SGS Labs themselves have a very excellent complete development platform for biosimilars, again offering an end-to-end solution to our clients. And an important aspect or a critical component in this end-to-end platform is the development of biological activity assays that are essential for demonstrating comparability, also essential for bioanalysis as the molecule moves further downstream. And so working with this, we set up a number of different assays, and I'm going to just touch upon the Bevacizumab assay again because it is much more relevant for this webinar. But we've worked with SGS now to qualify and test a number of different assays that we've developed here at DiscoverX. And so, as I indicated, the qualification exercise at Bevacizumab, with Bevacizumab, at SGS went off very well as expected, giving our partners an interrun accuracy of 115% and a precision of 12%, which is really incredible compared to what the HUVEC proliferation assay might have provided them. There's a lot of additional data behind this that, of course, in the interest of time, I'm going to skip over. But of course, if anybody is interested in hearing more about that, I'm happy to share that information with you. So far, I've talked about the use of this assay with regard to the Bevacizumab or Avastin molecule and how we've utilized this for validation, qualification exercises, etc., with just the Avastin molecule. However, there are two other molecules, as I mentioned earlier, that are anti-VEGF molecules on the market and high-profile biosimilar targets. And so the next couple of slides, I'm going to talk to you about some data that we've generated looking at qualification of these assays with Aflibercept and Ranibizumab. And as you would expect to see, that the assay performed very, very robustly with both Aflibercept and Ranibizumab, with excellent accuracy, precision, and linearity of the assays. And in each of these cases, we looked at two different analysts running these assays, running a range of 50 to 150% and getting good, robust data and a very strong, good linearity of the assay. So this is the data with Aflibercept, and we have a similar performance, again, leading to accuracy of 102% and precision of 4.4% and a linearity and a good linearity associated with this assay, again, with Ranibizumab or Lucentis as well. So altogether, what that indicates to you is that we've been able to develop an assay for anti-VEGF molecules that is based on the mechanism of action, has excellent accuracy, precision, and linearity, and is something that is now essentially a fit-for-purpose assay for use in lot release testing or looking at comparability between innovator molecules and biosimilar or biobetter molecules. The last point that I wanted to touch upon now is that all of this data that we've talked about so far and the entire assay, as I mentioned earlier, has been developed using cryopreserved, ready-to-use cells. And so the cryopreserved, ready-to-use cells actually allow us to go directly from a frozen vial of cells directly into the assay plate, run the assay without the need for any culture associated with that. And this approach has a tremendous number of benefits that it brings to the table, particularly in the space of QC lot release testing or in potency assay development. In particular, it significantly increases lab efficiency. So you can reduce, or in some cases, especially for particular assays, totally eliminate cell culture. And the amount of time that it actually saves you eventually works itself through a number of different calculations into about 0.3 FTE savings per assay per year. And so you can imagine that as you convert a number of your assays into cryopreserved ready-to-assay format, you can significantly increase the efficiency of your lab. And of course, as I mentioned earlier, it reduces your cycle time per assay, allowing you to have much greater flexibility where you can run the assay as and when needed for your samples, as opposed to having to batch samples, etc. The use of these frozen ready-to-use cells or cryopreserved ready-to-use cells also increase the consistency of assay performance. And that's actually really, really important because it completely eliminates culture-associated variability from the equation, where all of the cells that are frozen are cultured together, are all at the same passage number, are frozen at the same time, and essentially because of being treated exactly the same way, have a very, very consistent performance vial to vial. And that increased consistency in performance completely eliminates any culture-associated variability that one might observe from a day-to-day assay, especially if you're utilizing continuous culture cells. There's a tremendous cost reduction associated with that because as you save on cell culture and cell culture related media, the biggest cost factor in that is your need for the FBS or FBS substitutes. And it can save you a tremendous amount of time and money associated with that. And then finally, I think one of the big advantages of the system is that it allows for a very easy method transfer to global sites, particularly because you're looking at the transfer of frozen cell banks as opposed to cultured cells. And therefore, it makes it a lot easier for CROs, CMOs, or any of the global manufacturing sites to be able to easily take this assay on and start running it without having to deal with challenges associated with cell culture, particularly where you've got analyst-driven variability in cell culture, which can then affect assay performance or assay variability. So in summary, what we've developed now is a fit-for-purpose bioassay for anti-VEGF molecules, allowing us to assess the potency of these anti-VEGF molecules, as well as use of these assays for lot release testing and stability testing of these anti-VEGF molecules. As I mentioned earlier, the validation now is complete with Covance Labs, and we've also got additional data and additional qualification exercises done, not just at another partner CRO, SGS Labs, SGS Life Sciences, but also beyond that, now numerous clients globally that have used this assay very, very successfully. And we've also developed and tested this assay in a ready-to-use format, as I mentioned, in cryopreserved ready-to-assay cells. The last point that I want to touch upon here is that although we've talked solely about the anti-VEGF molecules and how we've developed this assay for anti-VEGF molecules, we've also kept an eye on the major biosimilar targets that a number of our clients have been pursuing globally, and we've developed over 30 different assays targeting these different anti-VEGF, biosimilar or biobeta targets. So here's a list of all of the different assays that we've developed for a variety of different drugs that we have developed, either assays that are already in cryopreserved ready-to-assay formats, what we call our bioassay kits, or They are also available as continuous culture cell lines where they can be utilized in a culture-dependent assay to develop a potency assay associated with these molecules. In addition to the targets that we've talked about so far for that are targets for biosimilar development, we also have a number of different targets, so over 750 different cell-based assays that have been developed at DiscoverX, and each of these can be adapted to create bioassay cells in terms of cryopreserved, ready-to-assay cells, or even the cell lines may be utilized to either develop your own potency assay or utilize this to develop your own banks for your own cryopreserved ready-to-assay cells. So this allows us to now develop bioassays for a number of innovative molecules apart from just the biosimilar molecules that I've talked about earlier today. So with that, I'd like to now thank you for your time and your attention on the webinar today. I'm happy to take questions. There's a lot of additional information that can be found on our website at discoverx.com/biosimilars, and I'm happy to address questions as they come in. If for any reason I'm unable to address any of these questions, or if you have questions that come to mind later, feel free to email those to me at my email address shown on the slide here, as well as once you get a copy of these slides, this will be available to you to utilize at that point as well. And if you have already started using our assays and you've got any technical questions, feel free to reach out to our tech support team. And if there are assays that you would like to see or that you would like to utilize that you don't see on our menu, we do have a custom assay development team that would be happy to work with you to custom develop any assays that you would like. So thank you again for your time and attention. I'm happy to address any questions as they keep coming in. So there's a number of questions that are already starting to come in. Please keep them coming, and I'm happy to address them. So the first question that's come in is, how do you manage stoichiometric expression of the two receptors on the cell surface? That's a great question. The way we approach this assay is actually looking at it from a functional standpoint. Our entire focus on these assays is to develop an assay that has the most appropriate functional performance as well as the most appropriate biology. So in each of these cases, the way we drive the development of this is that once we stably introduce both receptors into both constructs into a cell, we actually then start testing the cell pool for functional performance, then identify anywhere between 20 and 30 single cell clones that have been derived from dilution cloning, and then look for functional performance associated with each one of those clones and identify a number of clones that have the most appropriate pharmacology and the most appropriate functional performance. And that allows us then to really hone in on the end result, which is the functional performance, and bypass questions about stoichiometric expression, etc. Because at the end, the assumption that we make here is if the assay is performing as would be expected based on the pharmacology, we anticipate that the expression levels of the receptors on the surface will tend to be at a physiologically relevant level, or at least at a stoichiometric level between the two receptors. Another questions come in about the commercial availability of these assays. As I mentioned in the previous slide, all of these assays, so on the table on the left side of the screen, all of these assays have been developed as cryopreserved ready-to-assay cells and are available as cryopreserved ready-to-assay cells, as well as continuous culture cell lines, where it allows you to utilize these cell lines and culture them in your own facility and continue using them as long as you need to for these assays. On the table on the right side of the screen here, All of these assays are currently only available as continuous culture cell lines, and in a number of these cases, we are actively developing thaw-and-use bioassay kits for these clients. But in the absence of that, currently, we only have them available as continuous culture cells. Another question that's come in is, have customers utilized these assays for lot release testing of their molecules, and has this passed any regulatory guidance? Great question. So to address the first part of this question, we have numerous customers globally that are utilizing the Bevacizumab assay that we've talked about so far, and they're also utilizing a number of other assays that are on the list that I showed you previously, and they've utilized this at various different stages in their molecule development pipeline. The second half of that question is with regard to the regulatory acceptance of these assays. We've had to answer that question, I'm going to give a two-part answer. The first answer is going to be that we've had a number of our clients, as I mentioned earlier, that have been using these assays for lot release testing of their molecules, as well as a number of our clients that have gone through various checkpoints in the regulatory process, utilizing these assays. Again, all of those indicate that the FDA and the EMA authorities, as well as global authorities at various different countries worldwide, have at different levels accepted these assays and when utilized for lot release testing. In addition, we currently have a client that is using our assay for a marketed drug as well, and so we do know that it has gone through the full acceptance process. The second part of that answer is that we've also, in particular with regard to the Bevacizumab assay, as we've presented this assay at various different conferences globally, we've had a chance to discuss the application of the and the acceptability of these assays with the various different regulatory bodies. And what we've observed in each of those cases is that the person that we speak with oftentimes gives a very a straightforward answer in terms of these assays, of course, are based on the mechanism of action. However, whether any regulatory body accepts any assay is going to be dependent on the amount of evidence provided by the submitter. And so that's really the answer that we've received, and that's something that I think is a fair representation of what you can expect to hear from a regulatory authority, where with enough evidence provided, the regulatory authorities are amenable to accepting the assay that you might be utilizing for lot release testing of these molecules. Right, so if there's no additional questions, I would be happy to continue discussion with you at any point. If you get a question via email, I'm happy to also get in touch with you directly via phone or through any of our colleagues that are in the field, and discuss how we can get your team to start utilizing any of these assays in your facility. Again, additional information on these assays is available at discoverx.com/biosimilars. You have my email address on the screen here to continue any discussion or ask any questions that I may not have had a chance to address here. And with that, I will go ahead and end the webinar this morning. Thank you again for taking the time out to sign up and attend this webinar, and I look forward to continuing this discussion with you shortly thereafter. Thank you and have a great day. Bye-bye.